Hello everybody, in this video we're going to finish talking about the cell organelles. So this is where some of your classes left off. We were talking about ribosomes that make proteins. So for those of you who weren't in my class, you're going to hear about this. So ribosomes are kind of like little bunnies, right? In the summer you might find some wild bunnies just jumping around on people's grasses. But then you also have bunnies that live at home, right? I have a bunny at home. So ribosomes are kind of like that. Some ribosomes are just floating around inside the cell. They're traveling all over the place. But then other times you can find ribosomes attached to the endoplasmic reticulum, especially the rough ER. So that's what makes the rough ER rough, is because of those ribosomes that are on it. Okay. So if you think about it, it makes sense that ribosomes that make, if you have cells that are in charge of making tons of proteins, then you'll have a ton of ribosomes. Okay. Ribosomes are for all the organisms, because ribosomes make proteins, right? And all the organisms need proteins, prokaryotes, eukaryotes, all of them. Okay, moving on. So we have rough ER. Like I just said, the rough ER is rough because it has these ribosomes attached to it. So here's the thing. We have ribosomes that are just floating around, right? Those ribosomes will just make proteins that are used directly in the cell, right? It's kind of like you can make cupcakes, right? When you make cupcakes at home, where your mother makes cupcakes or whoever makes cupcakes, you can eat it directly at your house, right? You don't need to package it or anything. You just leave it inside a house, you eat it, right? Same thing with the proteins. If the ribosomes make certain types of proteins that need to get used directly inside the cell, they don't have a specific place that they're going, then those proteins can just be made by those free floating, those suspended freely, those types of ribosomes in the cytoplasm. However, sometimes ribosomes have to go somewhere, right? And if you have to have sometimes proteins have to go somewhere. So if you have proteins that need to go somewhere, now you need these ribosomes that are attached to the ER. So we'll look at some pictures, kind of make a little bit more sense to you, right? So here we go. Rough ER does have ribosomes. What they do is that they're no longer synthesizing the protein, they also transfer the protein. So when you look at this picture over here, rough ER is an example of a membrane-bound organelle. See all those membranes, right? So these membranes, they don't just form the rough ER itself, they can also bud off, right? So this part, for example, can just come off of the rough ER and form kind of a little bubble. We call that a vesicle. This bubble is not just an empty bubble. You will have, pretending this is a protein, it will have the protein inside of it, right? This is a good thing because now this vesicle can go to a specific place inside the cell. It can move on, right? It's kind of like if you make cupcakes at the house, but you want to give those cupcakes to someone else, you might want to put it in a little container, right? So these are the proteins that are getting shipped off to somewhere else. So rough ER is in charge of that. It will help transporting the material, especially proteins, inside the cell, and not only making the proteins. What type of organisms can have it? Plants and animals, or any kind of eukaryote can have it, because this is a membrane-bound organelle. And prokaryotes, such as bacteria and archaea, they do not have any membrane-bound organelle. Moving on, we have the smooth ER. So the smooth ER is smooth. So it should not say yes over here. I think I still haven't changed it, but it's supposed to say ribosomes attached. No. Okay, so please make sure you write down smooth ER. The reason why they're smooth is because they do not have ribosomes attached to it. So that means they're not in charge of making proteins. What do? They're in charge of two things. One, synthesize lipids. As an example, there are hormones that are made out of proteins, but there are also hormones that are made out of lipids. These lipids are made from the smooth ER. The second part is smooth ER can also detoxify the cell. So for older people who drink alcohol, for example, the smooth ER is what breaks down the alcohol and makes sure that your cell doesn't get poisoned, okay? So smooth ER can break down certain material, and as an example, it can break down toxins such as alcohol inside your cell. The smooth ER, you need to be able to see a picture and then recognize it, right? These are rough ER because they have ribosomes attached to it, but then the ones, they're still pretty close to the nucleus, but they don't have any ribosomes attached to it. It's a smooth surface. That is the smooth ER, okay? You're welcome to pause the video at any point to write down your notes. Moving on, we have Golgi. So the smooth ER, right? So the smooth ER It's also a membrane-bound organelle. You know where they are, right? They're only in plants and animal cells, not the prokaryotes. So make sure you circle the plants and animals. Moving on, we have the Golgi body, or the Golgi apparatus. The Golgi body is in charge of lots of things, but the most important thing is that it kind of tags proteins, right? So earlier we have a protein coming made from the rough ER. It gets into this little bubble called the vesicle. Then what's going to happen next is that this little vesicle will then fuse with the Golgi. It's kind of like two bubbles kind of becoming one, right? So this bubble will move to the Golgi, and then the membrane of this vesicle will become a part of the Golgi, okay? So then this Golgi will put some tags onto the protein, right? We have this protein. It's supposed to go somewhere. It might need to go out of the cell, or it might need to go to a specific part inside of the cell. But this protein on its own, it doesn't know where it's going, right? So what do? You have to add a little tag onto it. It's like adding an address to your envelope. Golgi is in charge of doing that, okay? So when you look at this function, Golgi is in charge of modifying, right? You modify by putting a little tag on it, that's modify. You can store the protein. You can package the protein. So once this protein is finished processing in the Golgi can put it into another, little vesicle and then send it off to somewhere else, either inside the cell or out of the cell, okay? So that's why it says Golgi function. Modify, stores, and package proteins and other materials from the ER, specifically the rough ER, for storage in the cell or release outside of the cell. So, but it's the protein. This material really is talking about proteins. Those proteins, they can either stay in the cell or they can go outside of the cell, okay? Who has it? Plants and animals both have it. This is a membrane-bound organelle, so they're not in prokaryotes, okay? All right, there's this video. I'm not really sure if you can hear it, but let me just talk you through this real quick. So let's take a look. Okay, so the endomembrane system, right? The endo means inside. Membrane are membranes, right? These are all membranes, right? The endomembrane system is talking about all these different organelles that have membranes. All right, see all these different parts? These are all a part of the endomembrane system because they're all made out of the membrane, okay? All right, see what happens over here? The rough ER makes the protein, but then it ships it off from using a little vesicle. But then this vesicle will move on to the Golgi, and then this vesicle becomes a part of the Golgi. But this vesicle is also made out of the rough ER. It's kind of strange, but right, so we see this little protein over here. This protein might need to go to the cell membrane and stay in the cell membrane. It might need to go out of the cell. It might need to stay in the cell. But either way, this Golgi needs to tell it where it's going, right? So it got these lipids. Let's see what happens next. All right, so in this case, we have these proteins, but then these proteins get packaged into a little vesicle, but this is a special vesicle. This is the lysosome. We'll talk about it. right? So if you just look at that, this Golgi can bud off a little vesicle, and then this vesicle can go to the cell membrane, right? So let's take a look at this together. Onto the next page, you can see that we have the nucleus, right? The nucleus is kind of connected to the rough ER. This ribosome over here makes the protein, but then the protein will get packaged into a little vesicle, right? Then this vesicle carrying the protein will go to the Golgi for further modification, right? You see these little tags being added to the proteins over here, right? So oftentimes these tags could be a carbohydrate, it could be a lipid, but it kind of, this chemical tag will help the protein recognize, but really it's just chemical interactions to tell where this protein is going to go next, right? So sometimes this vesicle can then come off from the Golgi carrying the protein that's already tagged into another vesicle, and it stays in the cell, or it can go out of the cell, or it can be become a part of the cell membrane, okay? So this entire process, where all these organelles, the nucleus, the rough ER, the smooth ER, the vesicle, a lysosome, for example, the Golgi, they're all a part of the endomembrane system. Did I say vesicle? Okay, all of those are a part of the endomembrane system, right? It's a system because all those components are always kind of fusing together, coming off from each other, that kind of thing, okay? All right, on to the next, we have the vacuole. Vacuole is in charge of water storage. So, right, as you can see on the right side, there's a picture of a plant cell. So this plant cell has a very large central vacuole, okay? It serves a few things. It holds water. It's kind of like a water tower. It holds water for the plants. Water is used during photosynthesis. Plants do lots of photosynthesis, right? So it has to have water. It has to have a supply of water at all times in order to survive. So the vacuole helps holding those water. Also, so it holds some salt and proteins and carbohydrate, really all kinds of macromolecules, but it also helps supporting the plant cell structure. It kind of keeps the plant cell kind of rigid, right? So if you've seen plants that are lacking water, the leaf kind of drop a little bit, right? That's because when you don't have as much water inside the vacuole, then the plant cell kind of, loses its shape a little bit, okay? This is only for plants most of the time, but animals sometimes can have many little vacuoles. The plant has one big central vacuole. This is where you should be focusing on right here, okay? All right, moving on to the next, we have a little bit more about vacuole. This one is called a contractile vacuole. What happens is, for a lot of prokaryotes, a challenge that they might have in life is that they might have too much water, because water naturally will just come into the cell, right? The cell content is very concentrated. There's a lot going on inside the cell. There are all those organelles and salts, right? So water will naturally go into the cell. That's a problem, because if you have too much water inside a cell, the cell might explode, right? So vacuole is made out of single membranes, so this is another membrane-bound organelle. So what happens with this contractile vacuole is, as you can see, it kind of is formed around water, and then it pumps the extra water out. An example is there's certain freshwater protists, such as paramecium and amoeba. They will form these contractile vacuoles. You kind of form this membrane around water, and then you get it out of the cell, basically. All right, on to the next, we have vesicles. So vesicle, we already mentioned. Those are little membrane bubbles, often surrounding proteins coming off from the ER and going to the Golgi, or coming off from the Golgi, staying in the cell, or going out of the cell. Keep going. So the vesicles are also membrane bound, which means they're only found in eukaryotes, plants and animals. And then vesicles, what do? They can store, right? They can keep certain proteins inside of it, and that's the end of the story. You just hold the proteins inside, right? They can store the proteins, store material, or move material between organelles, right? Those vesicle over here sends proteins from the ER to the Golgi, as well as to and from the cell surface, right? To and from the cell surface, okay? That is the vesicle. So a vesicle can also be formed when the cell membrane kind of caves in a little bit, and then you can form a vesicle inside from the cell membrane. You could do that, right? Or you can go out that way and then form a vesicle. You could do that as well. Lysosomes. Lysosome is a special kind of vesicle, but it contains digestive enzymes, right? Enzymes are made out of proteins, but these enzymes are real special. They're real strong. They're kind of like oh my gosh, lysosomes, lysol, right? They're all for digesting things, breaking things down. I wonder if those two words are related. Anyway, we have lysosomes that are mostly found in animal cells, right? It's a picture of a lysosome, but really it's a specialized vesicle with specialized enzymes in it. So it contains digestive enzymes. And how do you form these lysosomes? They also bud off from the Golgi apparatus, right? So you have the Golgi, you have those little sacs, right? But then you can form a little bubble off from the Golgi, and then if it contains specialized digestive enzymes, then they can act as a lysosome. So what they do is that they break down all different kinds of macromolecules, right? Sometimes you have proteins in your cell that your cell no longer needs. You want to recycle it, right? So you don't waste any material. So we have these macromolecules that can be broken down by the lysosome. But then lysosome can also break down kind of foreign invaders. For example, white blood cells have a lot of lysosomes, and what they will do is that they might take in a little bacteria, bring the bacteria inside the white blood cell, and then the lysosome and the bacteria will fuse. And then the digestive enzymes in the lysosome will break down the bacteria, okay? So I guess it's a good way to remember a lysosome is a Lysol. It can kill bacteria, right? It can break down all the different kinds of molecules, especially macromolecules. You remember macromolecules are massive, right? You break those bonds down, and now you only have monomers. And now those monomers inside the cell can be reused again. On to the next, we have cell wall, okay? Cell wall, as we talked about, is only for plant cells and is for bacteria and archaea, so prokaryotes. So cell wall for plants, and for prokaryotes, they provide a structure and it helps strengthening the cell. So the cell wall is more rigid than the cell membrane. It has some pores to allow transport of water and nutrient, right? So if you think about a plant cell, it needs water to come in, it needs nutrient to come in. So there's some pores in it. This part is not super important, so just take a look at it. Cell walls of fungi. So cell wall also exists for fungi. Chitin is the type of carbohydrate that is contained in the cell wall of fungi. But in comparison, in plants and algae, they have this carbohydrate called cellulose. Okay, so cellulose is a word that you need to know. It makes up the cell wall of plants. So what happens with herbivores, right? Goats, rabbits, cows, llama, they all eat plants for a living, right? The reason why this works so well for them is because they have enzymes and bacteria living inside their stomach that can break down the cellulose in the cell wall. So cellulose is a macromolecule of a carbohydrate. So then these herbivores, they have the ability of breaking down the cellulose so that they can get energy out of it, right? For us, for example, we're not herbivores, so we actually don't have the enzymes to break down cellulose. So when we eat plants, we're really just eating the plant fiber. We can't actually break down the plant fiber, which is why plants are good for us, right? It helps us digest. One of the reasons why plants are good for us. It helps us digest other food, right? So these cellulose, we don't digest it, and they're actually pretty difficult to break down. But anyway, so you really, all you have to know is cell walls of plants and algae are made out of cellulose. But I'm giving you a little more information on why cellulose is an important word for you to remember, okay? Who has it? Plants, animals, and some all the prokaryotes have it. All the plants and fungi has it. Some protists have it as well. On to the next, we have chloroplast. This is probably our last really important organelle. Chloroplast is in charge of photosynthesis. They're the location of photosynthesis. Next semester, we're going to talk more about how exactly chloroplast does photosynthesis. What can you see over here? There's an outer membrane and an inner membrane, right? Double membrane, right? Chloroplast, just like mitochondria, has its own DNA. It can self-replicate, has a double membrane. It is a part. Also went through the endosymbiotic process. What does it do? Performs photosynthesis. What happens in photosynthesis? Makes sugar using energy from the sunlight, right? So what actually happens during photosynthesis is plants will take water and take carbohydrate and do all kinds of processing with it, and then eventually make sugar. So we're turning inorganic molecules into organic molecules that contains a lot of energy. So what does it have? It contains chlorophyll, which is a pigment, but that's really where the photosynthesis happens. Chlorophyll live on these little pancake sacs. They live on the surface or the membrane of the thylakoid membrane. Okay, how many membranes? The outside membrane. We have two membranes, right? Two membranes that you should know. It has its own DNA and can self-replicate, just like mitochondria. Went through endosymbiosis again. Okay, if you're not sure what endosymbiosis is, please look it up on YouTube. Find a video, find a short one, watch it. I think that should help you make sense. But basically, the idea is we have a big prokaryote, we have some small prokaryote. They became one. At one point, the small prokaryote is not just living inside the big prokaryote anymore. Actually became an organelle for the big prokaryote over time. It's happened. Okay, it's quite the process. All right, almost done. Now we're just going to talk about movement real quick. We have flagella that are a long whip-like tail. This is how it whips. Okay, this is important. This is how it moves. Okay, flagella. They're made out of this thing called microtubules. We're going to take a look at that. Who has it? Prokaryotes, a lot of prokaryotes have it. Some eukaryotes have it, but not a whole lot of it. But it's for movement, okay? We also have another movement molecule organelle called cilia. So instead of a long whip-like tail, you get little tiny hairs all around the cell. And instead of going like this, they will go more like this. Yep. Kind of like how an octopus moves, right? So it just whips in one direction. It will not move too hard in this way, but then it will move really hard this way and then push the cell in a certain direction. So that's what cilia does. You have a lot of small short hairs that surround the cell. It's for movement. This is for prokaryotes or eukaryotes, just animals, because plant cells don't really need to move around all that much. Okay, Paramecium is an example of a protist that have these cilia. Look at this little tiny hair. Okay, so it goes all around the cell. Lastly, we have, well, second to last, we have pseudopods or pseudopodia. Pseudo means fake, podia means foot, fake foot. What is going on? We have these for animal cells. So what you see over here is that we have an amoeba. There's a little food. There's food. Okay, there's food. The amoeba will kind of extend a part of its body, but really it's just the cell membrane into a certain direction, drags the rest of its body over in this direction, and then surround the foods and take in the food and digest it. Okay? All right? Yep, so it just goes like that. But this is one big cell. This one big cell will extend in a certain direction, in the direction of food, and then surround the food. They become one, they digest the food. Okay? Cytoskeleton, I think this is our last one. Cytoskeleton is inside the cell. It's kind of this network of almost like train track inside the cell. Okay, if you think about train track, it connects all different parts of a city or a country, and trains get to move on the train tracks, right? There are lots of organelles inside the cell. Remember those vesicles that we were just talking about? How do the vesicles know where they're going, right? They have to follow some sort of direction. The cytoskeleton can kind of allow the vesicles to travel in a certain direction, right? And it also kind of keeps all the organelles where they need to be, right? That's why you don't see a nucleus all the way shifted to the end, or, you know, things like that. Functions. It maintains the cell shape, right? It's kind of like the people's skeleton, except this is cell skeleton. Maintains cell shape and locations of the organelles. It allows the transport of material within the cell. That's what I was talking about with the vesicles moving along the cytoskeleton. Assists cell division. We'll talk about that in the future when we talk about cell division. Assists cell movement. So with the pseudopods, the reason why the pseudopod can extend in a certain direction is because the cytoskeleton will move in that direction. There are three types: microtubules, microfilament, and intermediate filament. Okay, see the cell movement, all the cytoskeleton going in a certain direction, dragging the cell with it. Yep. For cell division, the cytoskeletons can attach to DNAs and split them apart, basically. But you don't have to know this, right? Just take a look at this picture. Know it exists. In the future, we'll learn more about it. There's a Kahoot that we'll play on Thursday. Okay. All right, have a good one, everybody. I hope that was helpful. I hope you took some notes. Try to remember it. Use the Quizlet.